Duncan Peyton is a venture capitalist slash biotech founder, having launched Aquarius Equity back in 2005 and more recently in 2014 founding 4D Pharma, where he currently serves as CEO. 4D is an interesting company on at least a couple of fronts. First, its flagship candidates are live biotherapeutics, a relatively new class of drug that we'll learn a bit more about as we talk with Duncan. And second, 4D's therapeutic endeavors are very wide-ranging. It's in or near the clinic in some cases, in some cases, multiple live biotherapeutic candidates in immuno-oncology, central nervous system disorders, respiratory disease, autoimmune indications, gastrointestinal disease, and even a vaccine platform. The company is on the edge of a new frontier of therapeutic innovation, or perhaps more accurately, in the gut of understanding how modulation of the microbiome impacts disease state. I'm Matt Pillar. This is the Business of Biotech, and on today's show, we're learning more about Duncan, his company, and its approach to developing and manufacturing live biotherapeutics. Duncan, welcome to the show.
Hey, Matt, how are you?
I'm good. Thanks for joining us.
Pleasure.
Hope I got the preamble kind of right there anyway. I mean, it's a super wide, diverse pipeline you guys are working on, and we're going to dig into that. I've got some questions for you on that, how you manage such a diverse and broad pipeline. But before we kind of move into that part, I have to tell you, in preparation for these podcast episodes, I do these weekly. I've talked with, I don't know how many, I think we've got 90 some published pods. I've got several more in queue. So I've talked with 100 plus biopharma execs, and in prep for them, I tend to go online. And when I went looking for background information on Duncan Peyton, I very quickly realized that this guy's like part of the 1% that doesn't have a LinkedIn profile. Couldn't find you on social media. Found a bit of a bio on your website. But I'm going to have to lean into you to tell us about yourself, because I can't tell my audience anything about you. You apparently shun the World Wide Web.
Yeah. I don't do I shun the World Wide Web? I mean, just sort of, I like to think that I'm focused on what I'm doing. I like to think I'm focused on, 4D, which is a really interesting, diversion into a new therapeutic modality. So, I want 4D science to do the talking, not my background, not what I've done in the past, not the fact that, I love rugby or, I think it's relevant. I think it's what's really relevant is, what we're doing at 4D and how that's going.
I can respect that. I'm translating that as, Matt, let's not dig too deep into my background.
No, I'm happy to answer questions. It's a bit of a weird combination of science, lawyer, banker, fund manager, and then, sure, ending up, running 4D Pharma, which, I will give you a bit of background. Alec and I, who I founded Aquarius Equity Partners with, we're doing all these, really, interesting orphan drugs, looking at medicines for juveniles. It was really good fun. We're doing a couple of investments a year, turned them over. We were pretty successful at it. But we always felt that, we were leaving, we used to see like 300 ideas a year. So we had a good idea of what was going on. And we always thinking, look, we want to find something where, we can build around it and, build a company and really take it forward, rather than, we had a drug, which is now called Fintepla, which has got approved for Dravet syndrome by Zogenix, which has now just been bought by UCB. Mm-hmm. That was a, typical example. We found some great data out in Belgium. We looked at it, we did some work around it. We sold it to Zogenix, and we held that for probably, 3.5 years. So you sort of leaving value on the table, and it was a fantastic drug then. It's a fantastic drug now, and we just wanted to do, build some therapeutics around the platform, and we came across the Microbiome back in, it was probably 2011 when we first started to invest in it. So yeah, that and I like rugby.
Yeah. Well, what inspired, you said you've got a legal background, you've got a VC finance background. What inspired life sciences? Like what's the life sciences inspiration? Did you study it? Did you kind of adopt it as you move?
No. My first degree is in biotech, so scientist at heart, really.
Oh, okay.
And you know what? It was really back, when I was doing what I over here called my O level. So when I'm about 16, and we're talking about biology, and then the biology teacher was really talking about genetic engineering. I thought it was really fascinating. I went on and studied biotechnology. Went into what was the real catalyst was. I went into a really small biotech, which ages me because this was back in the sort of early 90s when the first biotech revolution was going through in the UK. And I just thought it was just an amazing environment. We're developing some science. We went to list on the UK stock market. It was a really small team. I think it was the 4th or the 5th member of staff. So you got involved in everything. We listed on the stock market. We all went down to London, had a big party, listed on the stock exchange. And it was just a really fantastic place to work. Mm-hmm. Really great environment. You really felt you were pushing, science forward. And out of that process, I didn't have a PhD, and I knew I needed to do another qualification. And I worked pretty well with the lawyers. So I thought that was quite interesting. So I went back to college to, study law. Qualified as a corporate finance lawyer, focused in on private equity. One of our biggest clients was a large venture capital firm, and I went across there to as an investment manager to make healthcare investments, and then looked at what was going on. You work for a big firm. We had billions of dollars under management, and I thought, if you're looking at that space, there's a lot of great companies at a much earlier stage where you can invest, small amounts of money, and make a real difference to the pathway of that business. And I think that's what we set out to do at Aquarius, and that's what led us here.
Yeah. So 4D is a product born of Aquarius. Is that correct?
Pretty much. Yeah.
Yeah. Okay. How long? What would I say? 2014. 4D was founded.
Yeah, 2014 when we set the company up.
Was the company from its outset, was the company focused on the concept of live biotherapeutics?
So we had some projects running where we were investing in a microbiome project, and we just flipped that into a company.
Yeah. Okay. It seems like early days for that. And I want to, before we sort of talk about the chronology of the company's development path, let's just level set on what live biotherapeutics are. I think I mentioned to you, we've had some microbiome guys on the show. It's been some time, and we haven't covered it extensively. I know that the FDA sort of classifies it as a new modality, new therapeutic modality. But start up there, start kind of high and give us a rundown of what live biotherapeutics means.
Well, live biotherapeutics are defined as basically a live organism, which isn't a vaccine, which you can use, as a therapeutic, as a drug. So, for 4D pharma, that means we're using a single strain of bacteria that we understand the mechanism of, and that's really important. We understand why it's drug-like and how it impacts, the therapeutic pathways that we're interested in, and acts like a drug. And, it's really as simple as that. But also live biotherapeutics covers people who are using consortia, so like a whole batch of bacteria that are used against, for example, C. diff infections. You've got people who are using bacteria and engineering it to deliver a payload, a therapeutic payload into the gut. So that's what live biotherapeutics are. The simplest way of explaining it is, in some way, shape, or form, they're using something like bacteria as a therapeutic.
Yep. In your case, this may seem like it's a simple question, but it's one I hadn't contemplated until just now, just a minute ago. I thought to myself, how do you establish IP around a bacteria?
Yeah. Great question, right? You can't patent the sun. Isn't that what the sort of say? Natural existing organism, but actually, what you're doing is you're using these organisms, you're figuring out the therapeutic impact of them. You're figuring out, what molecules they're producing, what structures they've got that cause the effect that you're looking for, and you're using it as a therapeutic. Now, that's not how you think of a bacteria. You don't think of a bacteria as a therapeutic agent, do you? So you're taking it out of that context and putting it in a different context. And it was something we thought about long and hard. And there's a whole host of IP we've got around the strains that we're using, whether it be the therapeutic use, whether it be the structures, whether it be the metabolites that they produce that exert the therapeutic effect, whether it be part of the manufacturing process, whether it be part of the process that we use to discover these organisms, figure out how we built the platform to find out how these bacteria interact with the host. And that's where 4D Pharma has really got a strength. So we've got our own research. We don't license any research in. And that's led us to, I think we've got 1,500 granted patents now and similar number in in application. So, and that really comes from the science. This all started off in science, and figuring out what live biotherapeutics meant to 4D Pharma, and how to take them into the clinic, and figuring out all those pathways. So
Yeah, I'm gonna ask you some questions about that stuff you just mentioned, manufacturing in particular, and the science behind determining like what you're targeting and why. But give us a sense for, as I said, 2014 founding, even today in 2022, we're, I'd say, early days in this space. What do you think? Like, if you look at it's a difficult question to answer because we don't have a defined endpoint. It might be easy to define sort of a starting point where people started thinking, hey, the gut microbiome, we can draw a lot of connections between the bacterial consortium in our gut and how we're feeling and what diseases we're expressing and that kind of thing. But where would you kind of put us right now on the continuum? Is this, are we real early? Is this really like uncharted territory?
It's a good question.
I know it's sort of like I said, it's a hard question to answer. It's a big kind of vague question, but
like you go back to 2014, right? We think the microbiome, we could see a couple of bacteria that could impact, and what they were really doing was just damping down the immune system. Thought, well, that's pretty cool. Is it just these bacteria, or are there more bacteria? Is it the same pathway? Is it a different pathway? How does this all figure out? And that's actually what, led us to the thesis of, hey, look, these are impacting therapeutic pathways that we know about. So, why can't we use these as a drug? And then you start to unravel that and sort of think, well, is it just diseases of the gut? Is it just going to be things like IBS? Is it just going to be things like Crohn's? Or actually, does it have a systemic effect? Can we, use bacteria to influence the immune system to address rheumatoid arthritis? Can we use it for other diseases like that? And I think the big breakthrough came pretty quickly. I remember it was probably around about Christmas 2014. Al and I had just been across we're just flying back. I think we're flying back in from the States. We landed, and we got the first sort of set of readouts of strains that we thought had the potential to damp down the immune system. I think we were looking at asthma, rheumatoid arthritis, diabetes, and I can't remember what the fourth one was. And the initial results came back, and it was like, look, it hasn't worked. We're like, okay, and it was for, I can't remember what it was for. I think it was for diabetes. And then, but over that Christmas period, we started to get the results out of the other indications, and we actually started to see this positive trend. And you sort of thought, there's something here, and that's how we developed the platform to think about, right, okay, we can start, looking at systemic disease. And I think that's the real important point. I think a lot of people think the microbiome is just about diseases in the gut, and if you change the environment, if you change the makeup of the microbiome in your gut, you can eradicate disease. And I think you can if it's an infectious disease like C. diff, but when you need to impact the immune system, or you want to, modulate the gut-brain axis, I think you've got to be really clever and figure out the functionality that is drug-like. And I think that's what 4D's done. And, back in 2014, if you'd sort of said, hey, the FDA is going to grant you an IND for, Parkinson's disease using single strain orally delivered live biotherapeutics back in 2014, I would have sort of said, yeah, I think you're stretching it a little bit there. But actually, the science took us to that point. I think, the way that we got in the gut-brain axis was, as you're looking at anti-inflammatory diseases, you look at MS, and we started to see some really interesting neuronal differentiation. We sort of thought, we wonder if we can, impact the gut-brain axis, and that led us into all sorts of other stuff.
Yeah, indeed. I mean, that was one of the things that surprised me about the company. Is just, if you looked at it diagrammed out over a picture of the human body, you're all over the place, well beyond the gut. What about, just out of curiosity, super quick response on this? I know that other companies, biopharmas in this space, are pursuing an opportunity to, for lack of a better term or lack of better words than to simplify it, an opportunity to sort of reconstitute the gut microbiome after it's been ravaged by stays in the hospital, antibiotic, what have you.
C. diff.
Yeah.
Seal infections. Yeah.
That's a play for you as well.
No, that's using the microbiome, but in an entirely different way to how we use the microbiome. We use the microbiome as a targeted drug. Those guys are simply, trying to reseed the gut to grow a normal, question mark, what's normal, but a normal type microbiome. So it's really just outcompeting an infectious disease. Yeah. 4D is, as with drugs which target, different pathways, the immune system, the gut-brain axis, we are very focused on single-strain drug-like activity.
Okay. Walk us through the manufacturing paradigm there. What does manufacturing look like at 4D?
This is one of the things that we needed to figure out really, really early. You sort of think, right, okay, hey, this is great. We're seeing some really good responses. Actually, how do I get this into what does it look like? What do I give the clinician, to treat the patient with the disease that we got? And we needed to figure all that out. And, one of the benefits of single strain is, so we identify a strain using the MicroRx platform. It has a defined effect. We see that effect in the preclinical models. Then we start the development process, and it's like any other cell banking facility. You take, you have a master cell bank and a working cell bank, and you take a single cell out, you grow it up into a flask, which you then put into a larger scale fermenter. You then separate it down, freeze dry it, encapsulate it, and that's your product. And for a single strain, that's, it's a pretty, simple process. Mm-hmm. And we figured that out over time. And, back in 2014, we'd be talking to some outsourced manufacturers, and they'd go like, well, we're not really sure about this, and, how you do this and how you do that. And also their timeframes just didn't fit with how quickly we were pushing it forward. And that led us to, okay, let's find a site. Let's sort of think about how we can re-engineer it to our specification to allow us to, take control of our own manufacturing. And that's what we did. And there's a group of guys out in a place called Leon in Spain, which is northwest of Spain. Really, really beautiful city, fantastic cathedral, great people there. Mm-hmm. And bizarrely, they have a street called Antibiotic Avenue and Dr. Fleming Boulevard. And there was a city that grew up around, I think it was like mining originally, and then got into chemical engineers, got a great veterinarian school, great biotech school. And it ended up having the largest, I think, penicillin plant in Europe. Mm. So there's a great deal of, fermentation experience. And so we have the plant there, and, we're able to produce, reliable, reproducible, stable, drug product for, all our different therapeutic indications. I think what's interesting is, when we talk about oncology, when we talk about So MRX 518, which is our oncology program. When we talk about MRX 4DP004, which is our asthma program, or 5 or 29, which we're taking into Parkinson's. These are all different strains of bacteria. They all have different characteristics. Yeah. You know, like you or I, you know, you might like to eat steak, I might like to eat chicken. It's the same for bacteria. They're all different. They all have different idiosyncrasies about what environment they like to grow in, and how they're produced, and how labile they are to, you know, oxygen, air, water. And you know, we're just able to work through that. Great team. Love going out there. Haven't been able to get out there for a while, so looking forward to going back.
Yeah.
And just it's a good thing to have when you're pushing a new modality forward. You want to make sure that when you're talking to people, that you can sort of say, look, hey, look, you know, for us, you know, manufacturing is not an issue. We've got that ticked off. You want to go and see our plant? It's not a problem. You want to look at our shelf life? Not a problem. You want to look at our, you know, what's in there? You know, how much bacteria is in there? Not a problem. You know, monoculture. I like to sort of say, you know, we've been brewing beer for centuries, so it's a really simple process, but actually it's a bit more complex than that.
Sure. Yeah. No, I would imagine. So the process itself, though, from one strain to the next, the process and equipment required is relatively the same, I'm assuming, but it's sort of the, I don't know what you'd call them, excipients, perhaps. I don't know. Like, would it be?
It's the it's the media that you grow them in. So
that changes.
Yeah. So the media that changes, the temperature changes, the acidic conditions changes. You know, all these parameters in essentially the soup that you grow them in is specific for each individual strain.
Yeah.
And sometimes it's really easy. 518, the oncology program, you know, we got that right. We got that right really quickly, really stable quickly, got it up to the concentrations that we needed. You know, real straightforward process. 4 took a lot longer, just different bacteria, different criteria.
Mm-hmm. Yeah. How much space is required at this point, given your clinical candidates and what you're producing now? Big factory, or can you do it in relatively small footprint?
Yeah, it's a relatively small footprint. So we've got 3,500 liter tanks. So if you imagine, you know, like beer tanks, if you've ever been to a brewery, they're sort of, you know, similar size to that. And that's more than capable of supplying all our needs for clinical, and, you know, pretty much we can do production out of there as well. Yeah.
Yeah. Okay. You mentioned stability and shelf life. What does that look like?
Looks like 24 months at the minute, extending out. So we've got, you know, really nice stability. These are, you know, simple capsules that the patient takes, you know, twice in the morning, maybe twice in the afternoon, depending on what strain it happens to be. So from a patient compliance point of view, from a, you know, supplying the drug, it's pretty good.
Mm-hmm. Yeah. And that it's, okay. So what's your, I want to get a sense for that sort of administration paradigm. And I guess maybe the best way to examine that or take a look at it would be to talk about wherever you are most advanced in the clinic. So what's your most advanced clinical candidate?
So we have a program in IBS, which hopefully be moving into phase 3 study. And then we have oncology, which we've got a phase 2 ongoing. We've got a phase 2 ongoing in asthma. So happy to talk about, you know, out of those, I think, you know, talking about, you know, how people dose and how long people are on the drug. So, you know, the oncology program that we're running with MSD, this is in patients that have gone through numerous prior lines of therapy, have failed to respond. They've had a checkpoint inhibitor like Keytruda before that, and they've had a response, and then that response is tailed off, which happens in about 30% of the people that respond to Keytruda. Mm-hmm. And, you know, give these people Keytruda plus 518. What 518 allows the immune system to do, allows Keytruda to do is re-engage with the immune system, and you see a response. So we had people, you know, 2 years plus taking drug in that study, and, you know, seeing a response, you know, which is a great thing to do in terms of, you know, you've got patients who they've got no therapeutic options left. They go on Keytruda and your drug, and, you know, you give them an extra 2 years of life, which is just an amazing thing to do.
When you're striving to excel in a new arena, the best guides are the ones already doing it well. The Business of Biotech brings those voices forward to help new and emerging biopharmas turn their innovations, like mRNA and cell and gene therapies, into clinical realities. Tune in and subscribe for insights on hiring, regulatory, and other need-to-know topics for biopharma leaders. The podcast is brought to you in collaboration with Cytiva. Check out their resources at cytiva.com/emerging biotech. That's CYTIVA.com/emerging biotech. Tell me about any challenges that you'd faced in the manufacturing process around safety, toxicity perhaps. And it's this is a high level kind of, generalist question, because looking at it from a non-scientific perspective, you go, they're growing bacteria, they're putting it in a capsule, and they're administering it. And I know that everybody knows there's good bacteria and there's bad bacteria, but even too much good bacteria is not necessarily a good thing. So tell me about that. Like, what's the safety profile? How'd you go about figuring that out?
Yeah, so I think this is what drew us to the microbiome. Going back to 2011, you sort of thinking, well, look, if we're pulling these bacteria out of, someone who ostensibly looks healthy, if you administer it, it should be, that should give you a really good safety and tox profile. And the drug Fintella that we talked about before, fantastic drug, was repurposed, and it's higher doses, not the doses that they're using for Dravi. It did have, side effects and tox profile, which wasn't great. And if you can develop a therapeutic, never mind the modality, just a single therapeutic that is safe and well tolerated, it's really beneficial to the patients. The clinicians love it, especially if it's because we're using so many combination therapies these days. If you can bring something to a combination that has a beneficial effect and has no impact on safety and tox, then it's a really good thing. And that's what the microbiome, that's what single strain live biotherapeutics gives to you. That's what, our drugs, MRX-518, got a really clean safety and tox profile. We've dosed, I'm going to say it's close to nearly 600 patients. And it's a placebo-like safety and tox profile. And that is, to me, what's 4D-Pharma about? 4D-Pharma is about unlocking the power of the microbiome to develop safe and innovative new therapies for serious disease. That's what we talk about, right? Yeah. And that safety part is really important. And I think we're there. I think, in terms of developing safe drugs, IBS, placebo-like, cancer, oncology, placebo-like, asthma, placebo-like, so I think we've taken off that safety box. And I think we're a good way there on the, innovative therapies, Parkinson's, oncology, all these drugs are in the clinic.
Is part of that safety profile inherent in the delivery, the fact that it's orally delivered, and that our gut microbiome is well suited for safely handling bacteria? Is that part of the equation here?
I think we all, you say yourself, there's good bacteria and there's bad bacteria, and everyone understands how a single strain can change your phenotype, can change how you feel. Everyone's had some sort of travel illness. So same principle applies. You've got a single bacteria can impact you, make you feel ill. A single strain can help you get better, right? And I think, the microbiome, people assume, is there to help you digest food and do. It's not. It's developed over millennia. It's there for a reason. It has a really, we're starting to figure out, has a really profound impact on the immune system. We can change how the immune system works. We can rev it up, we can damp it down, we can impact, the gut-brain axis. So, if you think about how, the pharmaceutical industry is developed, what was it, 1937, I think the modern FDA evolved. We've got, 1,800 approved drugs, and we've got drugs that we've developed from the sap of birch trees. We've got drugs that we've developed from the fungus of a found in the Norwegian forest. We've got drugs that we're developing that are based around shark antibodies. Mm-hmm. And, what we do is we take all these products that we use very clever science for, and inject them directly, majority of times into the bloodstream, and then sort of wonder why we're seeing an adverse event, why your immune system's rejecting it.
Yeah.
And actually what we've never done is sort of think, what, we've got all these bacteria in your guts, which are in effect, fantastic little, drug development companies that have figured out, how they can reside in the guts and how they interact with the guts and, impact the immune system. And I think, that's the breakthrough, right?
Yeah. Yeah. Awesome. Yeah. I mean, I guess again, simple-minded. I'm a layman. I'm a guy from Pennsylvania who thinks simply, but why not introduce, effective, possibly efficacious therapeutics through the body the way that we normally consume and ingest?
We like to call it, you deliver them in the context that it's meant to be. So, the bacteria is meant to be in the gut and it's doing its job in the gut. Whereas, why don't we, for example, take the flagellum molecule from MRX518 and use that, develop that as a drug? Well, okay. So first of all, you've got to go through all the safety and tox issues that you face with a normal drug. Mm-hmm. And actually, when we have done experiments where we have isolated the flagellum and used it as a drug in preclinical models, it's not as effective as the whole cell bacteria. So there's something about the context of how these bacteria interact, i.e., in the state that they evolved in, that make them much more effective.
Yeah. Very cool. I want to learn about your regulatory experiences over the years, because as we've ascertained, this is a relatively new sort of frontier that's being explored. I'm assuming that as you set to work putting those manufacturing facilities together in the north of Spain, some of the stuff you were putting together was being seen for the first time potentially, right? And the sequences you were doing them, and there's some proprietary nature to how you were putting things together, and yet those sites need to be regulated, and the products need to be regulated. So, I guess I'd start just by asking you the open-ended question. Kind of share with us sort of how you would characterize your regulatory journey and the level of I guess acceptance and response you're seeing from the regulatory bodies like FDA and wherever else. I'm not sure where you're running clinical trials, so I don't know what other regulatory bodies you're working with. I know you've got some going on here in the States, so just kind of characterize that for us. What's it been like for 4D working with regulators?
Just being honest, it's been pretty straightforward.
Okay.
You go to them with a, hey, look, here's the scientific rationale and here's the data to back it up. Mm-hmm. Do you agree with our thesis or not? And if they agree with your thesis, then it's like, okay, you can go ahead. And you're right, they're asking questions that, what was good is they were asking questions that we'd asked ourselves, before coming to the FDA. And, they wanted to learn as well. And I think, both with the European regulators as well, we've helped carve that pathway through. Mm-hmm. But it's been good. They've engaged at a scientific level. They've helped us with our thought processes as well. I think people sort of think, oh, hey, it's a new modality. You've got all these issues to deal with. Actually, it's really nothing to see here. They've got their process. We worked through it, under a biologics application. It's been straightforward. The labs are all, CGMP. It's been really straightforward.
I guess it's a natural assumption that where you see something that doesn't have a whole lot of precedent, you assume that there would be some slowdowns, a lot of questions to be asked, a lot of things to be hammered out. But maybe where there are, maybe where there's less precedent, there's less obstacle too. I don't know.
It's just a lot simpler process, a lot more, a lot more understandable than, taking a piece of RNA, sticking it in a lipid molecule and then injecting it into someone which is a mRNA vaccine. It's a lot more understandable, and it's a lot more, straightforward. And the bacteria doesn't get absorbed into your body. The bacteria goes into the gut, does its job, moves on.
Yeah.
So,
cool.
It's a lot. We're doing something in the context, and we're just enhancing the functionality of the microbiome.
Yep. In the U.S., that your products are regulated by CBER, correct? With the FDA.
Yeah.
Okay. Yeah. Very good. So I want to give you an opportunity to talk a bit about your clinical journey. I look back and I'm like, wow, I think are you running 6 concurrent clinical trials right now in various stages?
Yeah, so we've got a fair amount going on within the oncology space, asthma, IBS, and now moving into Parkinson's. So yeah,
that's a lot. I mean, my questions would be my first question would be, how are you going about managing the execution of those 6 clinical trials simultaneously from a leader's perspective, from a corporate perspective? How are you managing that?
The whole process is, we need to get more data to prove out our thesis. So you want to be in a therapeutic area where you're stimulating the immune system, where you're showing that the microbiome can be used to stimulate the immune system. So that's what you want to do in oncology, working alongside checkpoint inhibitors or CAR T therapies.
Mm-hmm.
You want to be in an environment where you're sure you can use the microbiome to damp down the immune system, which is asthma. Mm-hmm. IBS was sort of a bit of a hybrid, because that was something that we started to work on very early to figure out manufacturing processes, etc. But nonetheless, really good data, which has helped us develop our processes, and then, gut-brain axis. And, all of these programs has inherent value to 4D Pharma, 518, 4 in asthma, etc. But what it's really showing is the value of the platform behind it, because to get to, the oncology, 518, to get to 4 in asthma, to get to, 5 in 29 in neurodegeneration. We've looked at rheumatoid arthritis, we looked at MS, we've looked at autism, we've looked at depression. So we really understand that the platform has a real, wide scope, and it's our job to show that. So it can look, well, how come you've got all these programs? And I just go back to it. This is where the science is taking us. Mm-hmm. We saw that, hey, look, you can actually stimulate the immune system rather than just damp it down. So you follow that path through, and that leads you to the oncology franchise that we've developed within 4D. So how is it managing it? Yeah, it's, we've just been through COVID. We managed to continue getting recruitment through. We got data out in asthma, which was great. Small number of patients, but we saw what we wanted to see. Safe, well tolerated, but really, saw an improvement in how those patients felt. How the breathing was, mm-hmm. Through the clinical scores. Less reliant on the inhalers. We're seeing. We saw the results, a lot earlier in oncology, where patients who we just didn't think were going to respond. I think the threshold in the study was like, we thought we might see 1 in 10 people respond. The last set of data, I think, we saw seen 4 out of 10. So you've seen a really good response rate there. So it's hard to juggle, but it's something that we need to do to continue to prove out the thesis of 4D Pharma. The unlocking the power of the microbiome, safe and innovative therapies, serious disease. We've got to get on and get it done.
Yeah, and that's where I was going with that. I mean, the point, the follow the science point is certainly not lost on me. But when you follow the science, and it's directing you into multiple good directions, you got to throw more resources at those opportunities. Do you I guess leverage or rely on lean into partnership to execute some of these things? And that may be a lead-in question, because I want to hear a little bit more about how you establish your partnership with Merck. Are they involved in the clinical trial aspect of that project?
Yeah. So we've got three areas where we're working with partners. So we have a trial with Keytruda in patients that are refractory to checkpoint inhibitors. So we're trying to re-stimulate the immune system in that environment. Then we have a trial with Merck KGaA or Merck Serono and Pfizer, and that is, we're just about to start that trial. The sites have just been initiated. We dosed in the first patient relatively soon, and that's in a maintenance setting alongside Bavencio for bladder cancer. And then we have another pure research collaboration with MSD, and that's using the MicroX platform to understand how we can develop the next generation of vaccines, i.e. how we can use the microbiome to stimulate the response to a vaccine. So they've all been involved. They're all involved in the clinical trials in different ways. In the research collaboration, Merck, that is really 4D Pharma bringing our understanding of the microbiome, and Merck, who are big player in the vaccine space, bringing that together to change how we think about vaccine and vaccine delivery. So that's how biotech companies grow, working alongside big pharma companies. It's been really good. I've really enjoyed working with Merck. I think they're a really smart company, innovative, love the science, and buy into it. And I think what those partnerships give to us, obviously, it helps us in terms of the development of those drugs and those drug programs. I mean, we couldn't afford to do the Keytruda study without Merck support. We couldn't afford to do the Bavencio study without Merck KGaA Serono. But what it gives to us is, it gives us, confidence that we're going the right direction. These guys wouldn't work with us if they thought, we were just, playing at it, and we're not. And it sort of validates the thesis. Why did Merck work with us? Well, Merck, have looked at the microbiome and, understand that, or they believe that single strain mechanistic functionality is the way to utilize the microbiome and where that play.
I talk with a lot of leaders, CEOs of emerging biopharmas, preclinical, who are still sort of making the plan, right? Still sort of going, okay, like in the long run, here's what we want to do. We either want to look for a partner, we want to look for an acquisition, we want to take the thing all the way to the finish line, we want to go public, right? We spend a lot of time on this podcast talking about those plans, and many in those early stages would can't even really fathom quite what it looks like to even partner with a company like Merck, as you guys have. So just give us, and we're running short on time here, I'm going to let you off the hook pretty quick, Duncan, but just give us some insight into when obviously you had good data, you knew what you knew about your product, but when you went out there and those partnerships were on the horizon, was it something that you muscled for? Was it something that sort of came to pass a little bit, a little bit more naturally? Tell us how those partnerships were established for 4D.
The way that it worked was, we were, you go into conferences, you're talking to a whole host of guys, and we met these guys from Merck, and just sort of said, hey, look, we think we've got something really neat that we think could work alongside, Keytruda. And it was really that initial conversation that developed, and I'd love to say it was like over a period of weeks. It wasn't. It was over a period of like months. As they got to work and look inside 4D Pharma, look under the bonnet, look at what we wanted to do, and then define how they wanted to how they would like to see the product work, and what sort of therapeutic setting those guys were interested in. And it took, you use the word muscle. I don't think it was really, maybe it was exercising our intellectual muscles in terms of sort of saying, we had answers to a lot of the questions that those guys asked, and there were some really tough questions. But it just goes to show the power of the platform, the power of the thesis, that we got to grow and, delivering on.
Yeah. Yeah. Well, it certainly is an exciting space. What else did you want to talk about? Anything else you want to share about what's going on at 4D, Duncan, before I let you off the hook? Yeah, I warned you prior to our conversation that I take a meandering path through an interview, so I may have skipped a stop or two. I want to give you an opportunity there.
I think one of the things about the microbiome is, it's a pretty broad church. It's, you asked me why I got involved in biotech. And I was telling the story about genetic engineering back in 86. Fascinating possibilities back then.
Yeah.
And if you think about the microbiome, it's pretty much like in that sort of vein. We don't think of CAR T therapies or mRNA vaccines or gene therapies as the same therapeutic modality. But yet the microbiome, whether you're using consortia, whether you're using engineered strains, whether you're using metabolites, whether you're using single strains like 40 pharma, the microbiome is all the microbiome. And it's not, it's the different approaches are very different, and the challenges that they face are very different.
Yeah.
And not one size fits all, in the microbiome space. And I think, that's a really important point that we need to start, to recognize. And I think, the guys that are looking at consortia, I think they're going to succeed in C. diff without a doubt. They definitely should. FMT works, so fecal transplantation works in C. diff. So there's no reason why that shouldn't. And as we progress through, we're generating more data to further prove out the theory of single strains to develop safe and innovative therapy. So I think one of the interesting things about gene engineering, like 1986, trying to think, was it 2012 was the first gene therapy, I think, which then got taken back off the market. Then 2017 when we started to see it. So like almost a generation. I don't think we're going to have to wait that long, for the microbiome to get an approved drug. And I think 40 pharma's going to be up there in terms of the first live biotherapeutic to address systemic disease, and that that's what we want to do. We want to bring these drugs to the marketplace, to the clinicians, to really improve the diseases that we're talking. I think you look at Parkinson's, devastating illness, devastating for the patients, devastating for the families. And if we can develop something that you can give early on because of the safety aspect of it, and you can impact that disease, that's just it's a gift. It's a gift. It's a gift to be able to be part of that story. Which is what drives us forward. So, yeah.
Yeah, it's exciting times. Quick follow up to the points you just made around, the distinction of what you're doing, addressing systemic disease.
Mm-hmm.
Does that become a problem? Is it at all a problem, like establishing that differentiation from the C. diff crowd? I've had conversations with the C. diff crowd, and I've asked them, like at a sub level almost. If you look at it visually, I've asked them if it's challenging for them to differentiate what they're doing from the freaking probiotic crowd, right? Where you go to GNC and buy a probiotic, right? Like, and to some degree, I've gotten, like we're playing on a different plane. You're yet a different plane. So is it important that distinction be created moving forward, or is it like more just public perception that doesn't necessarily concern high science?
I think there has to be a differentiation between one approach where we are just trying to outcompete an infection, to a very targeted approach where you are trying to address systemic disease, and they're entirely different. Mm-hmm. Approaches with entirely different science, and thesis behind it. And I think the breadth of disease areas that you can address using single strains, using powered by the platform that we've got, looking at how those single strains, the functionality of them, how it impacts the host cells. I think that's hugely different to what we see in trying to recreate what is a healthy gut. And I think the more that people see that we can use single strains as a drug, as a therapeutic to target specific diseases, then that's going to be interesting in terms of how we transform medicine going forward.
Right. Yeah, and hopefully that differentiation plays itself out. Becomes more understood. Perfect. Well, good. It's been a pleasure talking with you, Duncan. I appreciate the time you've given us. Again, subjecting yourself to my meandering line of questions. I appreciate it.
No problem. It's been an absolute pleasure.
Great. So that's 4D Pharma's Duncan Peyton. I'm Matt Pillar, and this is the Business of Biotech. We're produced by Bioprocess Online in partnership with Cytiva, which demonstrates its commitment to new biopharma innovation at its virtual biotech accelerator at cytiva.com/emerging biotech. Check that out. Check us out at bioprocessonline.com. Sign up for my newsletter while you're there. And if you're enjoying the pod, please subscribe, leave us a review, give us some feedback. And as always, thanks for listening.